My name is Vincent. I'm a PhD student from Leipzig, Germany at the Institute of Medical Informatics, Statistics and Epidemiology, and at SCATS AI. The project I present is conducted together with University Hospital Charité, our project partners. And yeah, I'm very happy to give you a short overview of what we're going to present in Lyon. So in general, we're interested in cross-species single-cell RNA sequencing analysis. And in this specific case, we investigate humans and hamsters in COVID-19. The special focus here lies in a disease state matching between humans and hamsters. And yeah, in general, the pipeline we're developing here is also applicable to other settings, diseases, but we focus here on hamster and human comparison. So for hamster data, we have two different species, Syrian hamster and Roborovsky giraffe hamster, where Syrian hamster has a rather moderate COVID course from clinical perspective, whereas the Roborovsky giraffe hamster rather has a severe COVID infection, COVID course. And for the human data, we use a publicly available COVID-19 cohort. So what are the disease states in the setting? For the human data, we simply have the WHO scale, where 0 corresponds to control group or no COVID, and 7 is the most severe COVID degree or corresponds to a severe COVID degree. For hamster data, it's a bit more tricky. So we have, as disease states, the days post-infection. Day 0 corresponds to the control group, and then we have day 2, day 3, day 5, and day 14. For instance, for our post-infection as disease states for the Syrian hamster, and we have day 2 and day 3 for the Roborovsky giraffe hamster. We basically perform two methodological ways. So on the one hand, we have a classical single-cell RNA sequencing analysis with a standard integration, and then followed by differential gene expression or pathway analysis. And I just want to give you a very short overview of how the integration looks like. So we started with an unintegrated object. We have obviously clustering according to species. And then we have our preprocessing pipeline, which is a very tedious approach. And then finally, we come up with an integrated object where we have clustering according to cell types and not according to species anymore. And this is especially important to have a uniform cell type annotation and to make sure that the species batches are not predominant. But how do we do the actual disease state mapping? So for this, we use a variational autoencoder pipeline or model. And we do this for each species, which means for each hamster species separately. So we learn one model for Syrian hamster jointly with human data, and one autoencoder model for Roborovsky giraffe hamster jointly with human data. And we do this for all cell types. So we start in high-dimensional gene expression space, learn variational autoencoder model, which embeds our data in a 10-dimensional latent space. And in order to get rid of the species shift in latent space, we compute what we call a species shift based on the controls of humans and hamsters in this latent space. And the reason we do this is that we want to avoid that the species or the control for the species also affects the infection responses. So that's why we only use the control data. And here on the down left, you can see an example how this then looks applied to real data. So on the left, we have hamster data. On the right, we have the human data and latent space or human representation before shifting. And on the right-hand side, we have this after the shift, and we can see that the human and hamster controls clustered together and the human and hamster infection cases clustered together. And in order to obtain our final mapping, we apply diffusion pseudotime distance metric to this shifted latent space data object. And yeah, this is a graph-based distance measure, and we take the mean diffusion pseudotime distance between the different hamster disease states and human disease states, and end up with heat maps or similarity heat maps for all cell types of interest, which are 8 in our case, and for both hamster species. So here we have the Syrian hamster and we have the Roborowski dwarf hamster. And what we learned from this mapping is, for instance, that for most cell types, the autoencoder model maps the hamster disease states to moderate COVID disease states. In the Syrian hamster, the same holds for the Roborowski dwarf hamster, but for instance, for neutrophiles, we see that there's a mapping of the hamster disease states to severe COVID human disease states, which reflects a bit, that the Roborowski dwarf hamster is used as a model for severe COVID. So this results we then in the next step want to compare with differential gene expression and pathway analysis to also get the biological perspective into this analysis. And I'm very happy if you're interested and would like to talk to me at the conference in Lyon. Thank you very much.